So my name is Greg Neely. I'm a professor at the University of Sydney, and I'm working at the Charles Perkins Center within the School of Life and Environmental Science. And what we do in our research is functional genomics. We basically test the human genome and try and understand how it works. Snake bites in general are kind of a major untreated tropical disease. So when you add all snake bites together, and basically about 5 million people get bitten each year. Of those, 2.5 million have enough venom to hurt them. And of those, about 140,000 is estimated die. And then there's another 400,000 that are kind of permanently disabled. So there's anti-venom, but it costs 7 times. You have to pay for it yourself if you get bit, and it costs 7 times your annual income to get it. And in some places, it's not even worth it for drug companies to sell it, so they discontinued it. So in some places there just really isn't any option. So with our work, we found a way to block the venom at the site where you get bit. So basically what we do is we use CRISPR gene editing, and we turn off every gene in the human genome, like in a big pool of cells where each cell is missing one gene. And then we add the venom to the cells, and the venom normally kills the cells, but we look for cells that can no longer die when they're exposed to the venom. And that tells us how the venom acts on our cells. So we did that, and we found a bunch of new genes, receptors and stuff. We also found a pathway that's conserved through all animals. And we used that to make a new antidote for cobra venom. And then that's what this study's about. When you get bit by a cobra, it causes local damage at the site of the biting, at the envenoming, and then it also causes like systemic damages. So it attacks your heart and your brain, and then you die. And so basically, the current antidotes or the antivenoms for cobra, they block the systemic effects, so they can stop you from dying, but you still get this terrible wound where you were bitten, and then that leads to people getting lifelong injuries and amputations, and kind of destroys their life. So our advance here in this study is that we made an antidote that can act locally, so you can inject it at the site of the bite, and it'll stop that wound that we can't treat otherwise. Within the venom, we found these toxins. They're called three-finger toxins, and that's what's actually killing the cells and causing the necrosis. And there are no drugs available that can block these compounds. So our antidote is blocking the cobra venom, but it's blocking it also through a new mechanism that we haven't been able to do before. So that's kind of cool. And so we can also look through all the different venoms in the world that have these three-finger toxins, and probably predict which venoms we can block. So Viper doesn't have them, and our antidote doesn't work for that. So that's another step we have to do next. For systematically testing all the different bad venoms we can get our hands on. And what we're finding is they use a limited number of mechanisms. So for example, this cobra venom mechanism, the blue bottle jellyfish in Australia uses a similar mechanism. And so we can also block that. We're developing that next. And so basically it's like a house. There's a limited number of ways into the house. And so we can block the front door or the window or the chimney. And so there's not that many different ways that a deadly thing can get into our bodies and interact with our cells. Our long-term goal is basically we want to make a small number of antidotes that block a large number of poisonous animals. And then we can kind of create like EpiPen type situation, where if you're in an area where it has a risk of these snakes or whatever animals that are venomous, then you can have this kind of one size fits all antidote. That's our long-term goal. So we're just going systematically through the deadly things that we can work on, and then collecting the mechanisms, which is not that many.